Good morning, everyone, and welcome to the final day of our Spark Summer Protein Crystallography collaboration for 2024. Just a few things that we're going to go over today. We have three things. One is I'm going to just do a quick tour of the beam lines and just give everybody an overview of the three main beam lines that we use at B&L and what some of them look like. Then Sharon's going to talk to us all about how the students transform themselves into becoming scientific users and the transition that needs to occur with the lab. And then we're going to talk about what's up for next school year. And we're going to lay out the game plan for everybody and talk about the different proteins we have and, give everybody a good sense of where we're going. And then that's it. And we'll see everybody in 2 weeks when school starts up. So yesterday, Kevin talked about beam lines, and he talked about specifically how the x-rays, when they come down, the pipe going down to the beam line, how those x-rays get focused and how the beam is intensified so that we can get precision when we're trying to collect data. And so the beam lines that we use at NSLS2 are broken down into a group called the MX Beam Lines. And of those MX Beam Lines, we use three of them. One that we've used in the past is the AMX Beam Line, which is, and they all have ID numbers. This is known as 17 ID1. And this basically allows us to collect a lot of data from a large number of samples with relatively large unit cells. What's important with this beam line is, and I highlighted it down here, is the spot size. This is 7 by 5 square micrometers. So that's the size of the spot that you have. So this beam line is really ideal for average to large size crystals. If we have very small crystals, we actually go right next door to the next beam line, which is FMX, which gives us the ability to look at some smaller crystals. The AMX beam line, this is the setup that they have. This is the robot. They call it Kermit because it's green. But right here is this thing here is called a goniometer, and that's what actually has the sample mounted, and that articulates the sample in the beam line. And the arrow that I have here, the red arrow, indicates the beam coming out. So that would show the x-rays passing through the sample, and then here's the detector, which the diffraction would hit, and that's where we would collect all the information. The robot pulls the samples from this giant dewar over here, and this dewar basically is a big reservoir. It's like a cooler that's filled with liquid nitrogen, and that's where our samples go in. That can hold up to 384 samples, which is a lot of samples. And what'll happen is this robot will pick each sample up and mount it so that we don't have to go into the hutch, take the sample off, put it on, close the hutch, re-energize everything. And it just saves a lot more time because you're using the robot to do all the picking of the samples and mounting and removing, and it just increases the ability of what we need to do and reduces the time. This is what it looks like from the workstation at the end station of the beam line. So again, there's Kermit. And then right here, this picture here, you can actually see this is where the beam actually comes out right this way. And this is called a beam stop, but the sample would be mounted right here where my little green pointer is. FMX has a much smaller spot size, and this is ideal for microcrystals that have a really variable size. It's also good for crystals that are often found in groups like clusters, or they have a needle-like structure. You can see the spot size here. It can range from 10 times 10 square micrometers down to 1 to 1.5. So you can have a really narrow, very narrow, tiny beam size, which is perfect for hitting these very small crystals. And we may have some small crystals that we'll use for FMX. And so that's the idea of using this beam line. Again, very small crystals, needles, clusters, it's ideal for that. FMX, again, this is a close-up of one of the samples in the beam. This has a different robot that's yellow, and they call that Big Bird. And again, for FMX, we utilize this one. It has the same features mechanically that AMX has. Again, the difference is that this has a more precise beam size. So what they've done with this one is they've used some more focusing mechanisms to help narrow that beam and get it to a much smaller size so that you can use this specifically for smaller crystals. And then the other one that we use more frequently is NYX, which is Kevin's beam line. And this allows us to do a wide range of experiments. We can hit some small crystals, but not as small as you would for on FMX. But it does give us the ability to look at some anomalous diffraction. He has an updated robot here. So he actually just got a new robot last year. I think they got a new detector the year before. So he's got some really well updated material there, which we can utilize. And again, this is what his end station looks like. Those are the computers that are there. This is a little bit more of a close-up of it. This is from when we were at one of the last beam times. So students will actually be working these computers. And there's also another one on the other side here, which you can't see. And then the hutch is just to the right side of this. And that's what that robot looks like up close. The detector is right here. This is the doer. You can see the steam coming up. That's the liquid nitrogen. And so this is the robot with the gripper that pulls the samples out and it puts it in. I was actually outside the hutch taking a picture through the little window because the beam was running at that time. And lastly, just want to show you a quick video of what a beam line looks like. This is a real quick video. So I'm going to just pause and share. There we go. Okay. But basically what this shows is, excuse me, this silver pipe here. Can everybody see the video first off?
Yep, we're good.
Okay, good. This is where the beam actually enters the hutch. So these walls, these white walls of this hutch are made of lead, which prevents the X-rays from coming through. So the X-ray beam enters here and it goes through a whole bunch of focusing mechanisms. So there are some, what Kevin talked about yesterday, the mirrors, and the reflectors. Those are all the focusing mechanisms that are in there. So you can see right here, here's one of the focusing components. Then the beam goes through here. It looks like these are some magnets in here that help to slow it down and focus it again. And then over here is another area. Then in here, I believe this is where the monochromator is, and that's going to help filter out certain wavelengths and gets the X-ray to the wavelength specifically that we want. Some more focusing mechanisms here, another monochromator, and you can see that this is the tube. Again, this is flexible, so I guess this flexes up and down. But the X-rays are moving through here. This is all under a vacuum. So again, inside of these big tanks are those mirrors that are doing all the reflecting. And again, needs to be highly automated so that you can make minor adjustments. And again, this is where the beam comes through this little tube here. Comes down here again, some more magnets, it looks like. And there's a scientist in the way. And then it continues down this tube, right? And again, this is a lead tube, and this will go all the way down through this focusing component here, some more magnets here, and then this will actually go into another hutch on the other side. And on the other side of this will be the hutch where the detector is and everything. So this whole hutch here that we're looking at is really just an entire structure that's made specifically to help focus the beam and filter it and get it to the precise diameter, energy level that we want. And then it goes through the wall here and it goes to where the detector is. So you can kind of see there's a lot of equipment in here. And this is a very long hutch. There's some teachers there too. And so again, that hutch is where we have all the focusing equipment and everything. And it's a lot of money to have that. I think something on the order of like $16 million is what it was. I think the last time Kevin had mentioned the cost. So it's a lot. There's a lot of equipment there. And it's a really neat thing. And when you guys are at the lab, you'll see that. You'll be able to see the hutches. So there are two hutches. There's the experimental hutch, which is where the detector is. And then there's the hutch that I just showed you here, which is where all the focusing mechanisms are. And these are big. These are gigantic buildings. I mean, they're inside the NSLS2. It's easily the size of like, you know, a kitchen and a dining room put together, you know. So you've got a huge room there. It's like, you know, a 20 by 40 room. So in some cases, it's almost the size of a classroom. All right. So that's it for the tour of the beam lines. Next, we're going to turn this over to Sharon, who's going to explain how we can get you to those beam lines and what students will need to do that. And then we'll talk about what's next. So I'm going to stop sharing and turn it over to Sharon.
All right. Sharon is sharing. Okay. Can everybody see that?
Yes.
Okay, good.
No.
No?
I just see up. Now it's there. Okay.
Okay. So, as Mr. Boland mentioned, I want to just walk you through what it takes to be able to come on site to Brookhaven National Laboratory so you can access all of these wonderful facilities and beam lines that you've been hearing about for the past 2 weeks. So, Brookhaven National Laboratory welcomes thousands of users and visitors every single year. And this really helps us to communicate our science to the general public and also advance science around the world and science at the lab that's been conducted. But again, these user facilities are open to anybody who's conducting research who could further the research with these particular facilities. However, BNL is a secure federal facility. So you cannot just walk on site and start accessing these user facilities. So what does this mean for you as a student participant in the Spark program? Basically, it takes some time and effort to be able to get through the BNL gates. And as long as you stay on top of the information that we are sending you, you should have no problem. We'll provide you with all of the information that you need to be able to access the Brookhaven National Laboratory. But it's when people tend to get delayed or don't complete everything that's needed is usually when there's some trouble. So there are things that you need to be able to come on site. This is going to include a registration, and I'll go through each of these in more detail, but you will need to complete a guest registration. There are required documents that you and perhaps your parents will need to return. You will have to thoroughly complete training that is assigned for you to be able to go into these unique spaces. You will have to receive a badge to come on site. And of course, we are very big into safety, so there are certain safety rules that you will have to follow. So first is registration. Early in September, we will send an email out to your teachers who will provide you with the information on how to go through the registration process. So once we know who wants to participate in Spark for the upcoming school year, you'll get an email that'll take you step by step through what you need to do to create this guest registration through our Guest User and Visitor Center, or GOV. So if you hear us refer to the GOV Center a lot, that is the Guest User and Visitor Center at BNL. The other thing that complicates this a bit is, so I am in the Office of Educational Programs, but to register you to come on site, it is a collaboration between our office, the Gov Center, and the NSLS2. So there are some things that we cannot do on our end that can only be done by Gov. There are some things that can only be done by NSLS2. So when people reach out to me saying, oh, I'm having a problem with this, but it's an issue that Gov has to deal with, you can see how it complicates things. So again, this is why we ask you to stay on top of the emails that we send out. So you'll create, you'll register through the system in the Gov Center. And once you complete your registration, this will provide you with a temporary GR, which is known as your guest registration number. Once your registration is fully complete, then you will be assigned an official guest number, or what we also call your life number. So anytime you come back to Brookhaven National Laboratory and you re-register to come on site for anything, you will be assigned that same guest number. So there are required documents for you to be able to come on site that you will need to provide both during your guest registration and when you first come on site. So you'll be contacted through the email that you use for registration to provide these required documents. Importantly, we strongly recommend that you do not use your school email address when you register. Most schools, not all, but most schools have a firewall in place that prevents you from receiving emails from Brookhaven National Laboratory. I don't know why, but it becomes a problem, and then everything gets caught in your spam filter, and this is unfortunate because then you're not getting the information. So register with a Gmail or register with something that is not your school email when you first do the guest registration. Some of these forms, if you are under the age of 18, may need to be signed by a parent or guardian. So these are minor forms that will be provided, essentially saying you receive permission to come on site to Brookhaven National Laboratory to conduct your research. You are also going to be required to provide documents that prove that you are a U.S. citizen or a legal permanent resident. That means green card holder. So you will be asked for this when you first do your guest registration. You will have to upload copies of these documents. You will also have to bring them with you on your first day on site at BNL to be able to receive your badge. So this can include, but is not necessarily limited to, things like your birth certificate and your high school ID. So those two things can be fine. If you have a license, you can bring that, or if you have a passport. So again, those three documents. If you only have your school ID, you will need to bring your birth certificate as well. So you'll upload those documents when you come on site. Make sure you bring the original document with you, not a copy. The Gov Center will not accept a copy of your birth certificate or a photo image of your license. They need the original document. Once all documents are uploaded and properly completed and submitted, then this is when you will be assigned your official guest number. If you are not a U.S. citizen, so if you are a legal permanent resident, it can take longer to process your guest registration. Don't worry, you can still participate in the program. It just may take a little bit longer, so maybe 30 days or more. You will also have to upload a curriculum vitae or CV or resume if you are not a U.S. citizen when you are doing that guest registration. We will provide you with a template that you can fill out to upload along with your documents, again, if you are not a U.S. citizen. Once this is complete, then and you have your guest number assigned, you will be required to complete training to access B&L. So there are different trainings that everyone coming on site to participate in the Spark program. I mean, there's certain trainings that everyone coming on site needs to complete, but additionally, trainings that you are participating in for the Spark program. So this will include things like, and these are all virtually conducted, so you can do these at home and complete them at home. In fact, you'll need to complete these before you come on site. So this includes things like guest site orientation, basically, what does it mean to be on site at B&L? Cybersecurity, so how can you safely access things like the Wi-Fi, what you should or shouldn't do when you are on site so that you don't open B&L up to cyber attacks. Excuse me, computer use agreement, very similar to the cybersecurity. There's an NSLS2 safety module because you will be accessing the experimental floor at the NSLS2. You need to know how to access it safely, so this will take you through that. Then there's General Employee Radiation Training, or GERT. So this one's a little bit trickier than the other ones. Importantly, when you complete this or the laboratory standards training, take your time. For all of these trainings, take your time because most of them do require you to complete a quiz at the end. The quiz can be anywhere from 10 to 15 to 20 questions, and it is required that you get an 80% on this quiz for you to pass the training. If you fail the training, it's okay. Complete it again, go through, take your time. If you fail it again, it will lock your training. You will need to reach out to us so we can request that your training be unlocked. If you fail a third time, it will lock your training so that we cannot unlock it, and you will need to go through remediation with the training staff on site at B&L. And usually it takes up to 2 weeks for the training staff to be able to schedule that. These are all courses that all of you should be able to pass. So this is nothing in here meant to trick you, but it is some, it is chemistry information, some of it is radiation information, all of which is provided to you in the training. But I know a lot of times when people are asked for complete trainings, what do? You just kind of click through, you click, get to the end. Oh, maybe there's some easy questions, or you just get the certificate. Not the case here. Make sure you're paying attention as you go through. Additionally, depending on the type of project you are conducting or the beamline you are accessing, you may be asked to require additional trainings. So for the protein crystallography group, that will include something like the cryogenic safety training. Basically, this means that if you complete this training, you can come on site and use things like liquid nitrogen to be able to fish for your crystals, which is important. So that's more for safety for hands-on. So again, and we will always tell you what trainings you need to complete, but it is your responsibility to complete them before you come on site. Okay, so arrival and badging. So when you are on site, it is required that you wear your badge at all times, and it must be worn. So you can see I've got my lanyard on. You can't see my badge right now, but my badge has to be above waist height, and I have to wear it at all times. We do have our own federal police on site, and federal police do go around, and if they see someone not wearing a badge, they will stop and question you. It is very important that you wear this at all times while you are on site. To be able to access B&L, The first time you come in, you will need a gate pass. This will be done by me. I'll reach out to you and ask who your driver is, or if you'll be driving yourself, to be able to come on site. If you have a senior license, you are welcome to drive yourself on site. We will give you a car placard that will allow you in. And once you have your badge, you'll show it to the security gate agents that you will need to stop and talk with when you first come on site. If you have a junior license, you cannot come on site and drive yourself with a junior license. It must be a senior license. If you are being dropped off, the person who is driving you must be a U.S. citizen. If your driver is not a U.S. citizen, so if you know that throughout the course of the year, you have maybe a parent or a relative who is going to be your consistent driver and dropping you off, and they are not a U.S. citizen, that is okay if you let us know as soon as you start the registration process, because then we can provide them with access to B&L that is linked to your guest registration. So you'll notice when we first ask if you're participating in the program, we will ask if your driver is a U.S. citizen. So we will start that process along with getting you on site. Otherwise, if we don't do it up front, your driver has to be a U.S. citizen when they come on site to drop you off. So you will visit the Gov Center, the Guest User Visitor Center, on your first day to confirm that everything that you need has been completed. So again, you'll bring the same documents. And again, we'll provide all this information to you again before you come on site. But you will provide the same documents that you uploaded when you first registered. And then if the Gov Center approves your registration, you will go and get your badge, and you will have that badge for the entire school year. So I think that goes until the end of June. So you will be able to come on site anytime with your badge. Safety. Safety is very important at B&L. It is our #1 priority. So your personal safety is of the utmost importance to Brookhaven National Laboratory. So we ask that you please respect all signs and instructions by B&L staff. We have never had a problem with this, with students coming on site. We don't expect to, but we just want to make sure that you are aware. When you're on site, you must follow all posted placards. So basically, if something's telling you you can't enter this area, you can't enter that area. It can actually cause huge problems if you go somewhere where you are not allowed. It can cause removal of your guest. You can be banned from coming on the site entirely, and it causes a lot of paperwork for me. I don't want that. Okay, so just make sure that you pay attention to these placards. We also want you to make sure you're wearing the appropriate PPE or personal protective equipment as needed when you are on site. So again, if you're coming on site to fish crystals, you're doing some wet lab work. If you were handling liquid nitrogen, you'll see during the training, you have to wear things like cotton gloves. And then over the cotton gloves, you'll have to wear latex gloves. And if you're handling anything with the liquid nitrogen directly, which most students won't, but you have to wear these other gloves. You have to wear a full lab coat. You have to have closed-toed shoes. So that's all the appropriate PPE. And again, you will be informed of that ahead of time. Just good point of reference though, anytime you come on site, make sure you're always wearing long pants and closed-toed shoes. That will get you through most of what you need to do. Everything else we can provide to you. Please do not attempt to use any equipment you are uncomfortable with or have not been trained to use. So again, you'll know we will train you on the NSLS2 equipment when you are there. Even if you've been trained and you're like, I'm not really sure if I, just stop and ask questions before you move forward, because we want to make sure that, first of all, you're handling it properly, but second, that you're collecting the correct data, right? So you don't want to, go through your entire process, collect data on all your crystals, and then realize you were collecting it improperly or didn't get the data that you needed. Additionally, if you are under the age of 18, you will be assigned a thermoluminescent dosimeter, or what we refer to as a TLD, which must be worn on the experimental floor at all times. So if you're coming to actually come on site to the NSLS2 and you will be on the experimental floor, such as at the NYX or AMX or FMX Beam Lines, you will wear this little, this big, it clips to your lanyard. Essentially, it measures whether or not you have been exposed to radiation. Has this ever been a problem at NSLS2? Never. But it is a security measure to know if there is, for some reason, a radiation leak and you were exposed, it lets them know how much exposure there was. So it's a safety measure that's in place. So if you are under the age of 18, you will be required to wear this at all times. But again, no worries, there has never been an issue. And there are a ton of fail-safes in place to ensure that doesn't happen. Okay, So that's what it takes to come on site. So just some expectations of you in participating in this program. So our expectation is that you are actively participating in every component of your research project. So this is from, project planning, experimental design and setup, sample prep, collecting the data at the beam line, doing the data analysis, and then presenting your research. All of this is really important, and all of this is a requirement of you in participating in this program. So we will early on ask that you provide a research plan. So this will be asked of all students or student groups that are participating. Essentially, it's going to focus your ideas and how you plan over the next, 6-8 months or so, how you plan on conducting your research project. We have your teachers and our office does every year, every couple years, we submit proposals that allow us to access the beam line. And those proposals are scored by scientists. So we have to submit proposals just like anyone else at who accesses the user facilities at B&L have to submit proposals. So what helps to drive the success of these proposals is your student research. So what have you been conducting? What have you found? All of this gets included in those proposals. So it's important that we have something to refer back to when you're moving forward with your project, so we know what it is that you did and how it turned out. So you will be doing lots of background research. As Mr. Bullen mentioned, you'll be talking shortly about the types of proteins and crystals you'll be working on over the next school year. You'll be conducting the sample preparation. What's nice about this is we do have collaborators, as we mentioned, that will be providing us the proteins that have been crystallized, which is one of the trickiest parts of this, but you will still be able to do that hands-on component just with something like lysozyme that crystallizes a little bit easier. So you'll still be going through all of the procedures. You are expected to be present for beam time. This is really important. So beam time gets assigned when it gets assigned. So we very often, the beamline scientists will reach out to us and say, we have these dates available. Your teachers will confer with our office and we will decide what the best date is. We will know this at least a month or two in advance of when the beam line date is assigned. So you will know well in advance when this is going to be. And the expectation is that you do come on site to collect data for your project. So this is very important. Just like you would make time for any of your other activities, you're expected to make time to come on site for this. For the crystallography group, this will tend to be on a weekend. So we usually schedule these dates for a Saturday because it's a little bit easier for people to be able to come. You don't have to worry about busing. You don't have to worry about missing school. Data analysis. So you're going to collect a lot of data at these beam lines. And then what do you do with that? So Mr. Bolin and Ms. Mackey already talked a little bit about things like COOT and Phoenix, and how do you take that raw data that you get at the beam line and make it become a crystal structure? How do you get the structure out of that, that molecular structure out of it that you will then, hopefully publish to the PDB? So that is part of the data analysis component. So you'll learn that software, you'll learn that technology to identify your protein structure. Timely communication. So this is super important. As long as you are communicative with us and your teachers, generally, again, we have no problems. What ends up happening where we have issues is, like I said, if we ask for you to submit something for a guest registration and we don't hear back from you, if you tell us you're going to be available for Beamline, you don't show up, and then we're texting you, calling you, trying to figure out where you are, and you don't contact us. So this is generally where we have the issues. So make sure that you communicate with us timely. And then you're also expected, like, again, any other scientists around the world, you're expected to share your data. And this can be done in a number of different and interesting and unique ways. So we have 2 meetings that usually occur around April or May in the school year. The first is the NSLS2 and CFN users meeting. So this is open to anybody who accesses and uses the NSLS2 and CFN user facilities throughout the year. They are invited back to present posters about their research. And there are other things that happen at this meeting, but generally the students will participate in this poster session, and sometimes are even invited to give presentations. So this is also a competition. So this poster session, it used to be, so you will be presenting alongside graduate students, postdocs, scientists from across the country who have come and used these user facilities. You'll be presenting your research. In the past, Spark students presented alongside or competed alongside these other individuals. Last year, they actually split the competition into general users and Spark users. So we had 3 winners in a Spark category of its own. I think they split that because the Spark students kept winning the competition. But it is a competition. It does come with some swag if you win and some certificates as well. We also have the Open Space Stewardship Program Symposium. So this is a symposium that is open to all students who have conducted some sort of environmental education, outreach event, activity, or research with B&L over the school year. So students are all invited to this symposium to present what they've worked on over the year. This is really nice. So you'll be amongst all of your peers and anybody who's come on site to do research at B&L. We have a keynote speaker. We have usually some food at the end. So this is a really nice experience too. We hope that you attend both. They're very different vibes and they're different audiences. You are expected to attend at least one of these symposia. Additionally, then there's publication. So we've talked a little bit about how you might publish your research. So this can be done through publication to the Protein Data Bank. This can be done through a peer-reviewed literature publication that could be into a scientific journal. There are journals that are also targeted to high school research that you can publish in. There are education journals, so even or methods journals. So there's different ways that you can publish your research. So that is another expectation, is coming here, how are you going to disseminate what you learned to the scientific community? And again, for our protein crystallography students, very often that is through these PDB publications. And there's a number of them from the past few years that have been published. So overall, what are your responsibilities? You submit those required documents and forms in a timely manner. Again, very important for non-US citizens, just because it can take a little bit longer to process yours. Importantly, you are responsible for this, for ensuring that all the documents are correct and up to date. So do not come on site with an expired license or an expired passport. This is the responsibility of you. It is not the responsibility of Brookhaven National Laboratory to identify this when it's uploaded. It's not the responsibility of your teacher to catch this. This is on you. If you expect any delays in your registration, let us know immediately. If you filled something out incorrectly, let us know immediately. The sooner we know, the easier it is for us to address or fix the issue. As mentioned, complete all required training prior to arrival, and you will receive all of this information. Again, you don't have to memorize it today. I just wanted to give you a feel of what's coming up in the fall. You are accountable for your project beginning to end. We want you to be safe, responsible, and courteous to all B&L employees and your peers when you are on site. And importantly, we want you to have fun. This is a really cool experience that very few people even get to experience, coming on site and using equipment at the NSLS2, let alone high school students. So this is really not something that is done at other national laboratories, or some maybe high school opportunities, but this is really unique that you have these facilities in your backyard. These facilities generally are not found at institutions of higher education or academic institutions, even corporate institutions, because they are so expensive. So you tend to find some of these things only at national laboratories. So this is a unique opportunity. All right, that's it from me.
All right. A lot of information, guys, that we will definitely go over again and again with you, especially with the registration process. And we get everybody through. So not to worry. We will make sure all the students have everything they need. Sharon is fantastic. She's really makes sure that everybody's up to date and has what they need. So this leads us to what's next. We have a lot of things planned for the year. And what's next? Well, the first thing that's going to happen is come September, you're going to start in your classes and your groups with your schools doing literature searches on the specific protein that each group will be working on. We will be looking for articles. We're going to review them. And we're going to read them and learn the background on the specific protein that our group is studying. We're going to use this information to help us develop a literature review. And the information that we get, we're going to share it within our groups. So we're going to share them not only in class, but we're also going to share them with the collaboration. We're going to set up monthly meetings, and then the partner schools can communicate with each other based upon their own schedules and how things work. But schools that are working on the same protein will share the information that they have. And ultimately, we're going to develop that literature review. We're going to get the background information on the protein. What is this protein that we're looking at? What is the bacteria that it is sourced from? What does this bacteria do? Most of these bacteria are going to be involved in some sort of infectious disease because we're getting them from the Seattle Structural Institute of Infectious Diseases. So it's going to have an implication on humans. So that's going to happen early in the fall. We're going to start developing that review. Along with that, we are going to assign the proteins to the schools. And so after careful consideration with the numbers of proteins and students and everything, ESM and Longwood are going to pair up with each other. And we're going to work on this dihydroorotate, the homodimeric type. Islip and West Babylon are going to work on the methylmalonic acid semialdehyde dehydrogenase. Wow. I need a breath after that one. And
I was going to say, we're definitely going to have to come up with some kind of a mnemonic for that.
Yeah, we're going to have to come up with
Nasty, I don't know.
Yeah. And then Hauppauge and Harborfields, you guys are going to work on the cyclophilin. What we included in here is the species it comes from. So you can use that information to begin your background research on some of these bacteria. And then we also have the ID number from Seattle. If you go to the Seattle website, you just copy and paste that ID number in, and you can get all the information about it. You can get the sequence, the actual amino acid sequence, where it came from, with the source organisms, all that. So there's a lot of important information on the Seattle website about how they actually purify this protein and where it came from. So those are the proteins that we'll work on. So the school districts here are going to all work with each other and develop their literature reviews and do some research on them. Now, each of these proteins is growing in multiple conditions. So within each of these proteins, there are going to be several conditions that we're going to see different types of crystals growing, and we may get different data sets. So there's a lot of information that's going to come out of what we're looking at. During this time, hopefully early in September, we'll get our registration information, and we will start getting all the students set up with their training and their B&L appointments. The goal is to try to get this done as soon as we can. While that's happening, we're also going to have students practice making trays of lysozyme, and teachers too, because we couldn't always use the practice. This includes pipetting the different volumes, changing ratios of the protein to the crystallization condition. We're going to practice harvesting with cryo loops. We have that there. Keeping accurate records of lab work and labeling the trays, understanding the importance of the metadata. That's probably one of them. Everything is important, but this is really important. We need to keep track of our samples, where they came from, what was the information that was in there. What's nice now is when we work with the University of Kansas, we get a spreadsheet with everything. So we have a tremendous amount of data that they provide, including a description of the crystals and how they felt the crystals looked and everything, as well as all the crystallization conditions. So that's something that we'll be doing in the fall. We're also going to learn how to use the computer refinement programs. We're going to try to set up some trainings using previous data sets so that students and teachers can become familiar with the process and eventually become proficient at these programs. I will tell you, and Kelly can attest to this too, you're never an expert. We're always learning. There's always something new. And so we'll try to set up some different group trainings and individual work trainings and provide updates so that everybody gets a good sense of how it works. And again, it's an ongoing process with this, but we'll start with the basics just to get everybody comfortable with these programs. We'd like to have monthly collaboration meetings, and have like a standing date. Like, for example, maybe like the second Wednesday of every month or something. I think once we get our school schedules figured out, we can definitely set that up. But we'd like to use this as time to discuss articles, maybe talk about some training on refinement programs. We might be able to set something up maybe at the lab in person, talk about our beam times, data collection, all that stuff. We're tentatively looking at beam time on NYX in either late November or early December. We have to work out some of the logistics with that, but at that point, everyone should have their training complete with their valid appointment and their badge, and the schedule is going to be determined. But this would be the first data set that we get on experimental proteins. And so at that point, we'll have some data that we can then look at and use Coot and Phoenix to help us with the refinement process. In the spring then, so we come back from winter break, Christmas break. We're going to continue working on data. So there's a lot of data analysis that'll happen. But then later on in the spring, everyone will present a poster for a presentation at the various meetings that Sharon had mentioned, the user meeting and the open space stewardship program. We're also going to have additional beam times. So we will be looking at beam times because, again, there are 3 cycles. So usually we'll get beam time in the fall. We'll try for some time in the winter, which would be like the February, March timeframe. And then again in the spring, right before school ends, in the May, June timeframe. And each time we collect data, there's going to be data to be analyzed. So we're going to do a lot of analysis, come that time in the spring. And again, we plan to have proteins crystallized and prepared for beam times at least all of these at some point. So at some point, we will be using NYX, FMX, and AMX. And again, everybody needs training in order to be to gain access to the experimental floor. So these are some students that were here last year and this year doing their work at the lab. And again, it's a great time. And this is at 2 different beam lines. So the top one is at NYX and the bottom one here is at FMX. And so that's what we have scheduled. So we'd like to open it up if anybody has any questions or if the teachers have anything you want to add, please go ahead. And students, the same thing. If you have any questions, go for it. Yeah, go ahead, Serenity.
I know you had said that you had to be 15 for the program and that if you were turning 15 soon, it was okay. But is there a certain deadline that we have to be that by?
What would that be, Sharon? That's your question.
We generally use the deadline as the registration deadline. So that'll be kind of mid to end of September. So that's usually what we use. If we need to, we can sometimes do another onboarding in January. But that depends on the number of students that we may need to onboard.
All right, thank you.
Okay. And Serenity, we could talk about that too in September when school starts up. We could figure stuff out. We can work out a plan. Anyone else have any questions, or teachers you want to add interject anything? We're good. All right, teachers, we'll hang on for a little bit after the meeting just to kind of go through the workflow again, make sure everybody's all up to date on it. But I think that's it for everything then. We want to thank all of our students and teachers for being here. We'll see you in September, which is a short time away. We're really excited about this. I think this is going to be incredibly successful this year because we have proteins that have crystallized. We know we have conditions that work. So I think we're going to hit the ground running and it's going to be a really successful year. So we're looking forward to it. Thank you everyone for your time in the morning and in the summer. We were hoping that 10 to 11 was pretty good. People were sort of getting up and getting going. And that's it. So we'll see everyone when school starts, and we will be reaching out regarding training and also regarding setting up collaboration meetings so we can get students and teachers alike to meet. So thanks everyone. Have a great rest of your summer. We'll see the students in September, and teachers will hang out for a few minutes just to kind of debrief and make sure everybody's all set.
Take care, everybody. Have a great start.